Vernalization. Lysenko, a Russian scientist, he worked on this concept and he found that cold requiring annuals and biennial plants can be made to flower in one growing season if one provide low temperature treatment to either young plants or moistened seeds. Lysenko is the first to coin the term that is vernalization. For the effect of chilling or cold treatment inducing flowering in treated plants. Vernalization is process of shortening the vegetative phase of plant and accelerating flowering by previous cold treatment. In many plants, flowering is not induced as proper photoperiod only, but they also require low temperature treatment for flowering. It means photoperiod and vernalization are two important processes responsible for flowering in plants. Low temperature or cold treatment given to seeds, also to bulbs or corms, promotes flowering in plants. On the basis of requirement of cold temperature, crop varieties are known as winter crops and summer crops. Biennials grow vegetatively in first season and will not flower until they are exposed to prolonged period of low temperature of winter. Many plants like wheat, rye, etc. require cold temperature of winter for flowering. One can define vernalization as acceleration of plant's ability to flower by chilling or cold or low temperature treatment. Simply one can say that vernalization is simple way as it is the process of inducing promoting flowering in plants by cold temperature. By vernalization, vegetative period is cut short, resulting into early flowering. Vernalization of dry seeds is not possible. For that, one has to soak seeds in water. We have vernalization effect. There are two important phases of vernalization. First, thermophase, which depends on temperature, and photophase, which depends on light. In any plant, if flowering is to take place, thermophase should occur over there and it should be followed by photophase. Scientist named as Melcher proposed that chilling treatment induces formation of vernalin, This hormone, vernalin, is transmitted to other parts of the plant to induce flowering. What is the site of vernalization? So, metabolically active meristems of shoot apex of mature stem or embryo of seed developing leaves and sometimes root apex are the sites of vernalization. Mechanism of vernalization. So to explain mechanism, we will see in brief some of the theories. So two theories are proposed. One is hypothesis of hormonal involvement and second hypothesis of phasic development. So first theory, hypothesis of hormonal involvement. Theory proposed by Chalukyan, which indicates that vernalin is produced in some biennials and perennials at low temperature. In long day plants, vernalin is converted to gibberellin. Anthocyanin is present in long day plants. This anthocyanin plus vernalin cause flowering in long day plants. But in short day plants, vernalin is not there and it is not converted into gibberellins. Vernalin is not converted into gibberellins as anthocyanin is not there in short day plants. Hence, flowering does not occur in short day plants. Second hypothesis relates with phasic development. Hypothesis of phasic development. It is proposed by Lysenko. Annual plants are having seeds and have series of phasic development which occur in predetermined sequence. Next phase only initiates on the termination of previous phase. There are two phases, thermophase and photophase. Thermophase, it is vegetative phase which requires low temperature, which ranges in between 0 to 14 degrees Celsius. And suitable moisture and aeration is necessary. This stage is of variable length and depends on nature of plant and environment. Photophase requires high temperature and depends on light. Here vernalin helps synthesis of fluorescein. And fluorescein is a stimulus for flowering. Thermophase should be followed by photophase. So see in this figure, A is some precursor. So due to chilling, B is produced and that is vernalin, D. And vernalin then induces formation of florization, and this florization then gets translocated where flower is to produce in the plant. So like this it is translocated. Now devernalization. So we have seen due to cold temperature at particular stage of the plant, in the seedling stage or at the embryonic stage, to the embryo, if one give cold temperature, then vegetative period gets shortened. This effect of vernalization can be reversed, and that process is the devernalization. So one can define devernalization as reversion of vernalization by high temperature treatment. It can be achieved by treating vernalized seeds or buds with high temperature. Devernalization should be done immediately after vernalization. Now applications of vernalization. Vernalization is helpful in shortening vegetative period of plant and bringing early flowering in many plants. Thus, there is saving of time to complete the life cycle. Early flowering induces early formation of seeds, which lead to early harvest, and this reduces the cost of cultivation, and it also protects the plant from unfavorable conditions if they are there in the nature in the latter stage of their life cycle, like drought or rain or maybe cold stress or maybe the attack of insect and pest, etc. Another application of vernalization is it increases yield and resistance to cold and diseases caused by fungi. Coronal wrinkle problem in triticales. It can be solved by using vernalization process. Vernalization can play crucial role in crop improvement. Vernalization is useful to temperate plants as well as tropical plants like cotton, rice, wheat or millets even. Now, the difference between photoperiodism and vernalization. So, we have idea of photoperiodism, and now we have discussed related with the vernalization, and now we will try to differentiate the things. Photoperiodism, we know it is a response of relative length of light and dark period. Vernalization, on the other hand, is acceleration of ability of flower by chilling treatment. In photoperiodism, stimulus is pursued by mature leaves, While vernalization, such stimulus is pursued by active meristems. Photoperiodism prepares the plant for flowering. Vernalization does not induce flowering but prepares the plant for flowering. Photoperiodism is mediated through florism. Vernalization is mediated through the vernally. Effect of photoperiodism cannot be reversed, but vernalized seed can be devernalized by giving high temperature exposure. Photoperiodic effect can be transmitted from one plant to the another plant through grafting, but vernalization stimulus cannot be passed from one plant to the another plant by such experiments.